Yes, Tracy.
So good afternoon and a warm welcome to all the dignitaries present here. I'm Srinji from Planet, serving as the elevated session assistant to ensure a seamless experience. Planet stands as India's most reliable digital platform, offering a multitude of emergency services exclusively for doctors. It is with great pride that Planet serves as the digital partner for this event, hosted by the Indian Society for Assisted Reproduction, Karnataka, and the focus of today's session is Microfluidics, how is it transforming reproductive technologies? So let's begin today's session, for which I would like to invite Dr. Ashwini ma'am. Over to you, ma'am. Kindly proceed.
Thank you, Tracy. Hi, all faculty, delegates, everybody. It's such a fantastic feeling to come back with the Journal Club. Today, we are here with the team from Santasa IVF, Hassan and Mysore, presenting on microfluidics. It's been a fantastic day today because it's raining in Bangalore. We have been waiting for these rains for a very long time, I must say. I hope it doesn't go to the extent of Dubai. We should have an exactly apt amount of rains, hopefully. It will chill us all down, hopefully. So it's a pleasant day. I welcome all the delegates who have joined us. At present, we have about 25 delegates. And here we are discussing on sperm selection techniques in IVF, of which microfluidics is a very important part. And the topic today is microfluidics, how is it transforming reproductive technologies? So I'll be introducing Dr. Soumya Dinesh. Can I have the slide of Dr. Soumya's introductory slide? Seema, you can share your screen, then it'll come. You're on mute, Seema. We can't hear you. Okay, anyways, to start, Dr. Soumya Dinesh, she is a fantastic friend. She's at present joint treasurer of KISAR. And
it's not showing normally. I'm done with you. This one? Yeah.
yes. Dr. Soumya, slide here, please. Seema, Dr. Soumya Dinesh slide ಗೆ change ಮಾಡಬೇಕು.
Let's make it slideshow.
ಅದನ್ನ slideshow ಮಾಡ್ತೀರಾ? So as you can see, Dr. Soumya has many degrees to her credit. She's an MBBS, MS, FMANS, FFM, FRM from Singapore, FRCOG from UK. She's a medical director of Santasa Fertility and IVF Institutes, Mysore and Hassan. She's also a medical director at SSM Multispeciality Hospital in Hassan. She's specialized in infertility and assisted reproduction techniques. She's a gynae endoscopic surgeon, and also she's proficient in ultrasound. She holds some very important positions. She is the secretary at Hassan Society of Obstetrics and Gynecology. She's an EC member of Kasuga and Kisar. She's a member of advisory committee for the Karnataka Art Board, which we discussed just a few days ago in the Kisar Connect group. She's a joint treasurer now at present in Kisar. She's joint secretary. And she's a very dear friend, a very lovely person. I invite you, Dr. Soumya, to take over from here. Dr. Nivedita will introduce Dr. Pankaj, who is the other moderator of today's session.
Thank you. Thank you. Seema, you'll have to share the screen. I will not be able to introduce Dr. Pankaj because I'm meeting him for the first time. So I do not know anything about him. So I need the slide. Seema, please share the screen.
I think I will only share the slides. Let me just, otherwise Seema is having some technical issues.
It's a good thing now. Participants also increasing, so it's good. We have a good.
Second, I'm sharing. Is this okay?
Yes, you can make it a slideshow if you can. That would be brilliant.
Is that fine?
That's perfect. Perfect. So Dr. Pankaj Kaingade is a PhD, a consultant embryologist, and a stem cell biologist. He's a founder and scientific director of Reprohelix Labs, Kolhapur. He's the scientific and laboratory director of Orchids Fertility Center, again Kolhapur, and is a senior consultant embryologist at Apollo Fertility, Solapur. He's the EC of ACE, Academy of Clinical Embryologists. He has a PhD from Gujarat, and his thesis is about reproductive stem cells with focus on endometrium and breast milk. He has several publications and he has edited and published several books. He's also a journal reviewer. He's got the Young Scientist Award from Charusat for research on breast milk stem cells. He's got the Indian Society ISAR Award for Manish Banka Young Achievers Embryologist Award in 2023 and the Dr. Gautam Kastugi's Young Embryologist Research Award for 2022. That's a very impressive CV. And thank you for agreeing to participate in our journal club. It's an honor. And over to Swami. Swami, you have to introduce your team and the talk. Yes.
Sure, I'll do that. One second. I will stop sharing the screen. A very good evening to everyone. I thank Kisar, Nivedita, Ashwini, all of you guys for giving this beautiful opportunity. And most importantly, this topic is very kind of, a little bit strange. So thanks for Pankaj for having accepted our invitation to moderate with me, because otherwise it would be all Greek and Latin, but I have tried to understand it to the extent possible in a couple of months. Thank you so much for accepting our invitation to join us as moderator. And Dr. Seema, she is our co-consultant in Santasa IVF, Mysore unit, and she's a DNB, and she has done postdoctoral fellowship in reproductive medicine with us only. So she's a very pleasant girl, very hardworking, but still has to learn a lot about screen sharing and computers. Over to you, Seema. Should I be sharing your screen as well?
Shall I share it, ma'am?
Yeah.
I'll just do that.
Do it. So till Seema starts sharing, Seema would be presenting a systematic review, microfluidic techniques, not only on sperm sorting, but also oocyte and embryo selection and sorting. So it's basically a complete overview of application of microfluidics in ART, like connecting to sperms, oocytes, embryos, everything. And even it goes up to the extent of doing non-invasive genetic analysis like NIPT. To that extent it goes. This particular systematic review is covering all those aspects. After that, I have a couple of slides to show you guys in a more tangible way how microfluidics is going to help us in the coming days. Then Pankaj would give us his insights, and then we will open question and answers.
Ma'am, is my screen is visible, ma'am?
No.
Yogita, can you please help her? No, otherwise I can share it from here.
Ma'am, here I can see the full screen, ma'am, actually.
No, Yogita will solve it.
Now it's working.
Yeah, it's working. Actually, I'm in Hassan. Otherwise, I would have taken over by this time. My hands are itching.
No way. We have already tried 2-3 times, ma'am. Everything was perfect, actually.
Just
No
once,
problem.
Yogita.
Participants are increasing. So it's good.
All right. Should I be doing it, Yogi? slideshow, Yogi. Unmute yourself.
Ma'am, is it okay? Is it visible, ma'am?
Yes,
Yeah.
yes. It's
Make
not
it
a
so-
slideshow, Seema. Yeah.
Am I,
Press the
am I
slideshow
audible,
button.
ma'am?
You're
Yeah,
audible.
you are audible, but make it slideshow. Go to the slideshow.
Ma'am, it is in slideshow, ma'am.
Okay.
Tracy, you want to see if there's any
Yeah,
problem
change
from
it
your
back,
side?
change it back, and then again go to slideshow.
It has come now, na?
Huh.
It
No,
has screen has
Yeah,
come
but-
now. Just make it full
Yeah.
screen.
That's what slideshow is not happening.
You can make it slideshow again, current slide.
Is it okay now? Is it okay, ma'am?
That's the one.
Perfect. Yeah.
Sorry, ma'am. Sorry for
Absolutely okay, Seema. Don't
my time
worry
for
about
this.
it. We all are very technically unsavvy, unlike Dr.
But
Soumya.
still we are trying to understand microfluidics. That's the challenge.
We can start. Yeah, thank you.
Very good afternoon, ma'am. Thank you so much for giving me this opportunity. Thank you so much, ma'am, and for everyone. And today is my topic we have selected is microfluidics. So this is the microfluidic chip as a promising evaluation method in assisted reproduction. It's a systematic review which has been taken from Bioengineering and Translation Medicine Journal, which has been accepted on 9th November 2023. What is microfluidics? It is a technological system that processes or manipulates small amount of fluids using microchannels. Aim of this journal is to understand the study design, to understand what is microfluidics, its application in ART, pros and cons. Is it okay, ma'am? Am I audible?
Yes, you are. Yes. You are.
Infertility is a global disorder which affects around 8 to 15% worldwide due to some environmental exposure, stress, lifestyle, and almost 2% of the childbirth nowadays is accounted for from ART. This is the conventional method where oocytes and sperm is taken out in vitro and it is incubated, and with the ICSI IVF, embryo has been formed and transferred to a uterus. But why microfluidics in this application? Because in the traditional method where there is the subjective judgment of embryologist is needed and it requires highly skilled and experienced assessors, it is time consuming, and the transferring samples between the dishes is very challenging. So microfluidics has a benefit to reduce the number of reagents, solvents, and samples. It lowers the risk of contamination and allows long-term culture and constant condition and automatic delivery with increasing its efficiency. So we have chosen this article because still now there were no focused studies on role of microfluidics in ART, but this systematic review has covered the role of microfluidics on gametes and embryo hardening. So far, we have a few studies on sperm sorting, biochemical analysis of the oocyte and embryo, cell manipulation, and drug screening and discovery. But this is the first systematic review which has been focused with all this relevant with the ART. The method they have chosen for this systematic review is like from journals, PubMed journals, Scopes, and Web of Science, and from PISMA protocol. And language has been not limited. They have taken from all the languages, and the data has been taken from 1997 to March 2023. With the eligibility criteria, having the inclusion criteria of having original articles rigorous with accessible peer review until March 2023, which has been the ART studies with the microfluidic platforms using cell or tissue from mammals. And they have excluded the review articles conference and meeting papers, computational simulation without cells or germ cells, observation which are not intended for ART and non-mammalian models. After this inclusion and exclusion criteria, the electronic database searches, they have searched around 5,340 papers, out of which they have screened the titles and abstracts and they've excluded. And finally, they have come with total 71 studies which have been included in this systematic review. And the publications they were taken maximum with 2020 and 2022 and least being below 2010. And these are few keywords they have considered for the study. Country-wise publications, the maximum number of studies they have taken from US, then comes China, Netherlands, Japan, and the samples they have taken were, 31%, 31.5% human and mouse sample, 19.2% from the bovine, and 16.4% from the porcine. These are the materials and the fabrications they have used, where PDMS that has been used in 49 studies of this article, and least being the polymethylacrylate. And the fabrication technology they have used, most commonly they have used is soft lithography. Coming to the sperm evaluation, around 38 articles were related to sperm in this systematic review, where 15 studies for human sperm and 26 studies were on sperm motility, and 5 studies they have taken from studied for sperm chemotaxis using oviducts, ovarian fragments, cumulus cells, acetylcholine, and progesterone. 5 studies were for sperm numbers, and there are other 7 studies were each for sperm impedance, applied whole genome analysis, its viability, beat frequency, opacity, and integrated interferometric phase microscopy. And this is the type, this is the sperm assessment, where to arrive the site of fertilization, the sperm has to undergo the rheotaxis, where it has to swim against the direction of flow direction, and this correlates the chromatin maturity. And the other one is the chemotaxis, where the movement is in response to chemical stimulus, where the sperm migrates along the concentration gradient in response to the chemicals. And the other one is the thermotaxis, where the movement of sperm is towards the source of heat. This is the microfluidic chip, where for the sperm analysis, where there is an inlet from this for the semen sample and also for the culture media, and the channel will be there. The direction flow, the flow of direction from the towards the down, and the healthy sperms will be flow against the flow direction and will be collecting in the collecting chamber. This is the other few microfluidic devices they have created, where they have mimicked, the first one has mimicked the female reproductive tract, genital tract, to assessing the sperm motility. And the other one, the second one is the microfluidic device that mimics the main reproductive tract component that is in the water, and the microscale groove which acts as a cervical surface. And the other one device is the probe with the angle of 20, 30, and 50 degree, which has been fabricated to form uterotubal junction and to trap the sperm. For the oocyte evaluation, there are around 20 studies in this review article, where 7 studied the mechanical property, 3 studied the compression force, 6 for the cellular membrane permeability, 4 studied the microspectrometry, 2 for the impedance, and 1 for the oxygen concentration. So this is to assess the mechanical property of the oocyte, where in the first picture, the pneumatic chamber has been used to create the pressure and the oocyte is observed under the microscope. And the second one is where the probe pressure has been given so that the oocyte will be pushed and the force sensor will be measured with the reaction force. So this is one of the this is one of the microchip device, where the oocyte will be fertilized, oocyte which will be used for the fertilization till the formation of the blastocyst. Normally, the culture media is held in the oil layer drops in a petri dish, and that will be manipulated by manual pipetting. But here in this microfluidic chip, microwell structure device integrates the single oocyte trapping, then the fertilization and embryo culture. So it can be cultured to blastocyst in this device. Then comes the embryo evaluation, where there are 13 studies in this review article for embryo evaluation. In this, 5 were for oxygen consumption was investigated. In one study, they have assessed the embryo mechanics, and one study for cellular apoptosis that assessed viability. 2 studies evaluated embryo structural alteration, and the other one for the real-time analysis of glucose metabolism, which was assessed in a single embryo level over 24 hours. So this is the microfluidic platform of embryo analysis, where the first, there will be concentration gradient of dissolved oxygen is formed by embryo metabolism, and it is in a hemispherical area. The second one, the B, is an electrochemical electrodes are used to estimate the in situ oxygen consumption. And the third one is a droplet with embryo and the magnetic beads merges with the reagent solution under magnetic force. These are other few devices where it has been used for culturing the embryo. The first one is a microfluidic device for positioning of the oocyte semen selection, in vitro fertilization and embryo culture. Here the oocytes are in 16 array microwell and has been corrected into 4 independent microchannels to introduce semen and the culture media. And there are other few microfluidic devices which have been used for other mammalian other than the human gametes. In this embryo evaluation, the triple metabolites, that is glucose, pyruvate, and lactate detection will be achieved within 5 minutes by serial measurement. The beta HCG was measured with the lowest sensitivity limit of 0.1 picogram per ml, and also the detection of tumor necrosis factor and interleukin 1 beta was achieved using digital microfluidics. Coming to the discussion part of this systematic review, they have many principal findings they have assessed. One is the sperm motility pattern, which has been controlled by microfluidic technology and mimicking the female reproductive tract microenvironment. Integrated microfluidic and advanced equipment, cellular mechanics, which has been related to reproductive performance of the oocyte and embryo, morphological or optical parameters to predict the gamete and embryo quality, and the oxygen consumption indicator for ART evaluation, and integration of microfluidics with artificial intelligence. And the other few studies about the fabrication materials and methods they have used. So far, sperm evaluation has benefit with microfluidics that it has stable concentration and temperature gradient, controllable flow direction and shear rate, and additionally we have been known about the chromosomal content, acrosomal integrity, and genomic alteration. And the benefit is it causes less DNA damage than the standard technique. And few studies have prepared a chip for human cervix chip, in which the in vivo assessment of sperm motility can be done. And there is the picture where the Zeal et al. Has been introduced by mimicking the female genital tract to assess the sperm motility. The drawback of microfluidic is the mechanical deformity, such as uterine peristalsis, could also affect the sperm motility, but no other chips have been reported yet. New point of care technology, where there is incorporation of smartphone based platforms with microfluidic chambers, which has been used to assess the sperm viability, DNA fragmentation, motility, and hyaluronic acid binding. The mechanical property of the oocyte has been studied by Alcasandra et al. Investigated the oocyte deformability by measuring the changes in the diameter and the surface area under 100 kilopascal compression, and he found that changes in the oplasm surface area amplitude were positively correlated with decreasing quality. Morphological and optical parameters. Here, the optical parameters have been proposed to assess the oocyte and its fertilization rate, and they have found that shorter wavelength and higher arbitrary unit were recorded in fertilized oocytes than the matured ones. And this is a non-invasive than the conventional method. Oxygen consumption indicator for ART evaluation. Here, the metabolic indicator can be used to reflect the embryo quality. Embryonic metabolic rate was positively associated with the following embryo transfer, and metabolism and oxygen consumption serves as an objective and quantitative parameter to evaluate the embryos. Microfluidic chip with built-in detectors of embryonic metabolism can be constructed or being constructed. So this is oxygen consumption spherical diffusion theory by Date et al. He has implemented an electrode array in the centrally positioned microwell, and he has demonstrated the increase in oxygen consumption from morula to blastocyst. And this embryo respiration monitoring can be sensed with a measurement within 1 minute. Advanced microfluidic chips. Here, the microfluidic chip can be merged with the machine learning algorithms like real-time cameras and electromagnetic devices, and also it enhances the CASA accuracy. The artificial intelligence makes prediction based on pattern recognition by incorporating the processing power of the computer. And this AI gives results with minimum human intervention, and it diminishes the intra and interoperative variabilities. Coming to the materials and methods for microfluidic fabrication, commonly used are organic materials like PDMS, PPMA, polyester, polyvinyl chloride, and polymethylene, where PDMS, that is polydimethyl siloxane, offers several unique and attractive features, that is non-toxic, affordable, optically transparent, and it is permeable to air and supports long-term culture. The fabrication method, most of them use photolithography, and here in this research study, most of them have used soft lithography. And there are other few called injection molding, laser ablation, etching, and 3D printing technology, which is low cost and environmental friendly. The limitation of microfluidics is there are few limitations they have elaborated in this study. More elaborative studies using human oocytes and embryos are needed to validate their safety, efficacy, and reproducibility. The lack of microfluidics popularization itself limits its use. And microfluidic chip might not be universally accepted by all doctors, who must be trained before using them. Coming to the conclusion part, microfluidics is a young but established technology of ART for its efficient and accuracy, objective evaluation of sperm, oocyte, and embryo than the traditional approach, artificial intelligence with microfluidics to assist in the daily activities for the embryologist. Microfluidic chip enable a precise and point-of-care pipeline to reproductive samples and to promote ART. And the last one is the novel cues are obtained on cell motility, morphological alteration, impedance secretion, oxygen consumption, and metabolism at single cell level for the gamete and embryo. The other information about the study is systematic review. The main author is Tongwu, and the funding financially, this systematic review study has been supported by National Key Research and Development Program of China and the Natural Science Foundation of China. There's no conflict of interest, and these are the few references I have taken. But for the systematic review, they have used around 152 references are there, and these are the few more. Thank you.
Thank you, Seema. It was a very quick and excellent presentation. hopefully all the postgraduates who are, who have listened to you would definitely get benefited, and hopefully there would be a question on microfluidics in that paper. So I will now I will share a couple of slides so that what Seema discussed so far it's like, it's all mostly it is theory, but also like I'm trying to make it more tangible with videos and actual photos of the microfluidic chips. So then maybe we will get a better understanding of that. I will share my screen.
Shall I stop sharing?
Yeah, you should stop sharing, Seema.
Yes, ma'am.
Did you stop sharing?
Yeah, yes, ma'am. I have stopped it.
Okay. So is it visible?
Yes, it is.
Yeah,
It's
Dr.
visible,
Samya, it's visible. Yes.
but do it slideshow.
Sorry?
Slideshow.
Yeah, I'm going. One
Yes.
second. Is it good now? Okay.
Yes.
So this is basically what a clinician should know. Not should know, it's always better if they know. So in what direction we are heading, especially those ART practitioners, like in the coming days, how easy IVF is going to be. So we know that IVF, I'm referring to IVF, ICSI all put together, ART, but since ART involves IUI, I'm using IVF in the place of ART. It's a nascent field and it's been there since only last 50 years. We are achieving what we want and also what couples want, but the end product, that's the baby or the maternal outcome, may be suboptimal, as we have all witnessed in many couples. It could have been much better, embryos could have been better, or the outcome could have been better. So gamete selection, in vitro culture condition, and even the protocols we are using to do embryo transfer, they need further optimization, and there is a lot of evidence pointing towards it. For example, recently we came to know that frozen embryo transfer is not optimum because there is no carpus luteum, and carpus luteum would have produced some bioactive molecules which would have aided in good placentation. That we got to know after many years of using frozen embryo transfer, because we missed out on a nice chunk of physiologically active element, that is carpus luteum. And it's common sense that what we are doing in IVF lab, we are missing out multiple chunks of such extremely effective interactions. For example, what happens to the embryo inside fallopian tubes, we are not able to replicate in our IVF labs because we are putting them into in the culture media, but we are not providing all the materials and substances that are secreted by the ciliary cells of fallopian tube. So all the nourishing that endometrial cells give, we are not able to give through our culture media, which appear very basic compared to what a body could have given. So our goal should be to mimic the exact physiology of female reproductive system, and microfluidics is effectively bridging that gap. So we have been hearing that for ease of use, microfluidics, we are using the terminology chip. So whenever we are saying chip, we have to assume that that is based on microfluidic technology. So there are organoids that the microfluidic chip has one cell type, maybe from uterus, endometrial cells, or from lung cells. Then it will function, that chip will function as lung. So we can understand what happens to lung cells with using that particular chip. So those are called organoids, and there are organs on a chip. So multiple elements when we use endometrial cells and the stromal cells, fibroblasts, mesenchymal cells together, and we also provide a vascular endothelial cells along with extracellular matrix, that is a component that is provided by bioengineering techniques. Then it becomes an organ on a chip. So the scientists have produced the entire flow of events that happens in ICSI, So we have ICSI on a chip and also human on a chip. So there are multiple organs on a chip connected through vascular channels, so that we know how liver responds to something secreted by the intestine, and what happens to pancreas when that particular substance gets into pancreas. So like that, scientists have been able to study complex interactions in these platforms. So we're coming to what is being gynecologist and fertility specialist. I'm focusing mostly on our organs: uterus, vagina, 3-4 organs that we have. So vagina on a chip, it's very simple. The donor vaginal cells from the donor are taken, and they are grown inside the channels, and there would be fluid containing nutrients as well as estrogen, so that vaginal cells proliferate well. And this has been used extensively to study vaginal microbiome and how lactobacillus versus other bacterial species how they affect the vaginal microenvironment. And they have also produced cervix on a chip. This chip has been used to understand how HPV induces carcinoma in cervical cells, and also they have produced a 3-dimensional cervical cancer tissue on a chip, so that they can use a particular new drug. Or in a patient, for example, we can out of 10 options to use for her, whatever drugs available, we can test those drugs on these chips from her own cervical cancer cells, the chip which has been made, and that would get exposed to those cells, those drugs, and whatever is showing the most efficacy, that drug can be chosen. It's almost similar to like culture sensitivity, like you grow that bacteria and you decide which antibiotic works well for that bacteria. In that similar lines, but in a much complicated way, we can use this technology. There are ovoids. So we know that in vitro maturation of oocytes came with a big bang, and then we thought, okay, for PCO women, this is the thing to do, because we are not stimulating, we are not putting them at the risk. Then we did collect immature oocytes. We tried to culture them further in IVF labs, but we added only the maximum thing that we could add was FSH, LH, and only hormones. But that is not enough, because compared to what happens inside a ovary, Graafian follicle, what we were trying to do was a very meager thing. So definitely the technology failed because the results were pathetic. So coming further, scientists have developed extracellular, whatever cells there in a Graafian follicle, they have incorporated on a chip. And there are two things: either we can grow immature oocytes in this platform to mature oocytes, and one step ahead, going ahead, we can use stem cells and induce them to grow in this particular fashion, and try to make them mature oocytes as well. And there are 3D printing technology is a very big boon for this particular microfluidic technology. The scientists have produced basically printed. That means they have printed the extracellular matrix, the supporting structure of an ovary, which contains vessels, the stroma, all that different architectural supportive structure. And they have seeded inside the vascular channels, they have seeded vascular endothelial cells. And in the stromal area, they have seeded fibroblasts, mesenchymal cells. And in key positions, they have seeded stem cells. So they have been able to produce ovaries as well as testes. Mind it is from stem cells. And there is one very beautiful technology, that is Avatar. It's almost similar to Avatar because it's like Avatar of female reproductive system, developed by Dr. Kim et al. This has vagina, cervix, uterus, fallopian tube, ovary, as well as liver. So all the organoids are connected to each other in a way that one affects the other, and we know that, okay, if you manipulate this particular cell, how will that affect the liver? And if you put something to the ovary, how does it affect the endometrium, and vice versa? So like this model has been used in developing drugs for cancers, cervical cancers, and even endometriosis. Body on a chip also, as already I discussed, it's very useful in drug discovery, drug development, and personalized medicine. Especially right now, it's being used mostly for cancers. Those who are interested in this particular technology, they can visit this Harvard University website, Wyss University website, and you would get to know more very interesting things. Actually, I was lost when I took this microfluidics topic, thinking, okay, let us do something new. Then as I kept on digging deeper, it was mind-blowing. And those who have some inclination for research, kindly go through this website. And I'm going to play some videos here. There is some beautiful technology called multi-electrode array chip. I'm going to skip and just how there are neurons in the brain with multiple different connections. We can actually, they have already produced, one second, sorry. They have produced microfluidic chips to the extent of they have placed microelectrodes that one electrode can stimulate one cell, like, to that precision. It not only stimulates, there are some holes inside through which they can add some molecules also. They can chemically also, they can change the stimulation. And see, basically we can see this particular dish with stem cells, and we can differentiate them into whatever cell type that we want. In this video, they're showing them differentiating into neuronal cells and dendritic cells. And this complex interaction, what happens between the cells, can also be registered through the signals, which is recorded through electrodes. And this is an amazing technology indeed. And there's one more dynamic chip, like am I audible?
Yes, you are. Yes.
Oh, thank you so much.
Yes.
Okay, so there is a beautiful model here. Some organs are dynamic also, like lungs and intestines. So they have made a microfluidic chip, and they have seeded. There are two layers. This is a permeable membrane. On top, they have seeded lung cells. Below, they have put capillary cells. So here, air will flow from the top chamber, and blood will go through this. And actually, it is not recorded in this video, but the side channels can be compressed. They can move through vacuum. So this particular model has been used to develop biomarkers for COPD, asthma, and also to develop newer drugs in those situations. Okay, this is our our bread and butter. So this is a very beautiful video. I hope all of you will watch it with keen interest. How easy it looks, but also it's an amazing thing. It's the size of a hand phone. But there are multiple chambers here. In this chamber, they are feeding cumulus complex. All OC6 are fed here, and they are held there, and then the sperms are fed, the prepared sperms are fed in this droplet. And there is a valve here. I don't know if you can see my cursor. that's operated through magnets. So it will release one into the next chamber, and then they would add hyaluronidase, and then there would be holding time. Once the cells get dispersed, there is suction, so they would suck out all the other things. It would come to the next level where they would assess whether it is mature or not. If not, that goes and gets loaded here. Again, they will assess. Then if it is not mature, M2, it comes here. If there is a polar body mature, then that goes to the ICSI chamber, and that is held there. Then this particular sperm, then they will release one sperm, and they would activate the sperm through laser. That's amazing. Rather than trying to catch the sperm with micro needles, that is easy. Then that just gets injected, and then that comes and gets, parked here for further culture. Isn't it beautiful?
Yes, it is. Yes.
One second. Yeah. So that's all. Thank you so much for your kind attention. I would stop sharing my screen. Okay. So now over to Pankaj.
Yeah.
We
So
are
first of
waiting
all,
to listen to you.
yeah. Actually, first of all, thank you, Kisar, for inviting me to moderate this session. And I thank Dr. Nilita ma'am, Soumya ma'am, and Yogita ma'am. Because of Yogita ma'am and Soumya ma'am, I am today here. And I'm really happy to be the part of this journal club. Actually, when I was given this topic, I was knowing that only sperms are being sorted here, the microfluidics. But when I read this paper, yes, they are talking about oocytes, they are talking about embryos also, right? So even I took 4-5 days to read all these papers, and I thank Dr. Seema also. She had very crisply, she had taken all the points from the papers, and it was very accurately demonstrated over there. So now anyway, Dr. Soumya ma'am and both of them have already spoken very much about this topic. But I would like to just share my views on this topic. Now, as you know, all these two papers came in 2023, and it was from the same author. The main author is Dr. Wang. He's from China. So as we know, most of this sperm, most of this technology in the wet lab they're using is mostly with sperm only. Oocyte and embryos are still, and some animal model studies are going on. So I would like to just give my perspective. Now I'll just go through the paper, what exactly they told. So as we know, they were dealing with the sperm examination, the oocyte evaluation, and embryo evaluation. So as we know, there are applications of microfluidics. They are into diagnostics also, drug delivery. Now, just Soumya ma'am had told many things about drug delivery regarding the stem cells of how these drugs are being delivered. I'll also give an example regarding this thing. And also cell and tissue engineering. Actually, this microfluidics is an area where we cover the subjects like physics, chemistry, biochemistry, or cell biology, or tissue engineering mostly. So when we deal with the diagnostics, microfluidic devices enable rapid, sensitive, and cost-effective diagnostic tests for a wide range of conditions. Then also drug delivery, microfluidic systems can precisely control the release of drugs, improving targeted therapeutic efficacies and reducing side effects. Also cell and tissue engineering field also. Now, again, as I told, in the sperm evaluation, they mostly studied the sperm motility, the concentration, then the independence, the visibility, the viability, the beat frequency. This all studies were done under sperm examination. And when it came to the oocyte evaluation, they studied the oocyte preparation or this thing. So in that, they morely studied the mechanical properties, the membrane viability of cryopreservation, the oxidation concentration. And when it came to the embryo evaluation, they studied the embryogenesis, mostly the oxygen consumption properties, embryo mechanics. Even they studied some when HCG concentration or some growth factors like human necrosis factor or interleukins. So these all studies are being done by using the microfluidics technology. Now, as Soumya ma'am had given some good videos and all these things, in that to that, I'll give just an example regarding the stem cells also. For example, when we do a PRP, the main aim of this PRP is to enrich the whatever the microenvironment with the growth factors and all this. So by using this microfluidics also, you can prepare all these growth factors or whatever microenvironment is there, and you can just load on a chip and you can go and deliver into the respective organ. So that is nothing but the drug delivery applications with respect to reproductive organs. So usually now this microfluidics is in all biological fields, and now recently it has entered in our reproductive system also. So basically they are trying with sperm, oocyte, and embryology. So basically what we read from this paper was that mostly all the studies are related to humans and mouse also. Maximum studies or wet lab or whatever technology we are using in lab right now in IVF lab is with sperm. Oocytes and embryos will take some time, but it will be there in the future. And why microfluidics? It is there because it is one of the cheapest mode to dwell with. So these are some of the applications or some of the uses of microfluidics in this field. So yeah, I'm done with this.
So shall we look if there are any questions?
There are a
Not
couple.
yet, I feel. Thank you so much for such a wonderful presentation. Thank you, Dr. Seema. That was very elaborate, very nice. Thank you, Dr. Soumya. It was really good. I saw the video. I think Palermo, way back in 1989, when ICSI was first propagated, the best thing that the scientist did was he didn't patent it. He went on teaching everybody, and that was a remarkable thing as a human. There was no commercial interest at all, and that made ICSI being available for all of us. And you know how it has remarkably changed the scenario for male factor infertility. I'm glad the costs are low because it's not patented. That's one thing. About 2 years ago, I think at ISHRE, I that was 30 years of ICSI. And I heard Dr. Palermo speak about ICSI, and I happened to see the same chip. He actually presented the chip that day. I was thinking that it may take 5 to 10 years for it to come into general lab. But do you know of any lab which is using it, or it's still in the fancies or dreams?
Scientific labs only. With
Not
scientists.
yet for clinical use. All right. I think at present the only thing that is for clinical use is the sperm sorter.
Yeah.
As Dr. Pankaj said. Yeah. So when do you see, sir, that we may start using microfluidics on the egg or the embryo? When do you foresee that can happen?
Maybe it will take some time, but a decade.
A decade? Oh, okay.
Yeah, maybe because it will take some time because there are some this thing. Yeah, it will take some time, but it should come.
Okay.
Even this technology is working from long time. But now this microfluidics, what we are using it is from just 5 to 6 years back.
Oh, great.
I'd like to say something here. I have sometimes very young the kids coming, want to do embryology and all that, and they come for a opinion. Do you think embryology should we join? And that kind of, I do get a lot of kids coming for that. And the fear is that soon we will not need any embryologists. The way microfluidic, everything is so automated. Should we even take up embryology? And I think that is not going to happen even in 5-10 years. It will take time, and the human touch is not going to be lost. It's like when artificial intelligence is coming, a lot of people are getting laid off, and human requirement is less. But I don't think embryologists are going to. It's not going to go out of fashion. You need the embryologist here.
So in microfluidics also, to load and unload, you need an embryologist.
Absolutely.
You need an embryologist. They are not kicking out embryologists. So it's like a no-touch technique with a nature's precision, I would say. So
more objective. It'll be more objective. The selection.
That's what, with nature's precision, with a human touch, and loading, unloading has to be done by.
Santosh, it is artificial intelligence. The sperm is selected by the machine.
No, but somebody has to load somewhere.
Nature's precision is not there. It is still by the chip.
They're trying to replicate it. I mean, they replicate the velocity, etc. So many factors which are in nature are replicated by the chip.
Yes,
like the speed, whatever.
See, even let's say in that chip you are loading sperm. So sperm will reach to the egg by the principle of microfluidics, which they believe in fallopian tube.
That way. Your mimicking nature is what you mean. Yeah. Okay.
That way.
Okay, good. Yeah.
Maybe eventually we're going to reach 100% success rate. What say Soumya? I feel microfluidics will give us 100% success rate. Then we can put up all those billboards.
Madam,
Yeah.
I read somewhere that it is predicted that 1954 we would be talking about 100% success rate to our
So,
patients.
I would like to add one point. It won't be 100%, but whatever the lifestyle which we are living today, now it is going to worsen day by day. So, so we need technologies to at least cope up with the whatever
Yeah,
rate which we are
100%.
getting it today now. Just to cope after a few years, we have to cope with this result also. So what I feel is that not 100%, but we have to cope with our lifestyle later on with these technologies.
Correct.
Possibly. I have a
think-
question to all the delegates who are here. Is anybody using the sperm sorting with microfluidic chips?
Can I say?
Yeah,
I'm
yeah,
using it.
Santosh, yeah.
I'm using it a lot and I'm finding it very useful.
Okay.
Okay,
Oh.
and I have used it in many of my previous IVF failure where we did not form embryos and all those things without, but there is something you need to change. So I changed the sperm sorting method and I found it very useful. And only problem with this, the count and motility should be to some extent reasonable.
True.
You cannot use it in
SOAT. Yeah.
So I have done quite a bit of cases and I feel it is useful.
Please tell us
But-
something about it. Which brand, which company, and how do you do it? Tell us something about it.
Is it that Zymot? That's the chip.
I'm using Zymot.
Zymot is there, Qualis is there, Lensuk is there.
Yeah, which also? Zymot and?
Lensuk, Zymot and Qualis. Yeah.
Okay. Great.
Is
Even
there
the
one
cheapest
brand?
method, even the cheapest method is that you can go for a double ejaculation also. So that will also give a good low DFI. What we have studied. I'm using both the things. Even if some patients we can't go for microfluidics, that time we go for a double ejaculations also.
Okay.
The main thing is to have to lower down your DFI. So microfluidics
Can
also
you
good
elaborate
option.
on the double ejaculation? I mean, how much time difference?
We take
between-
around 2 to 3 hours gap, and after that, but usually if the pickup OP is there in the morning at 9 o'clock, we usually tell the patient you can ejaculate the sterile container at your home and just drop it here also. If in case you don't have second ejaculation, we can use the same. Or also we have some backup sample also. So usually we'll tell you in the morning you have an ejaculation. After that you come to the clinic. So before the procedure, we'll take another sample. So usually that has given a good results. Even I have one paper with oligospermia also, where we had done a double ejaculations and that had given a good results for us. And
So, but
even-
you're not using the first one, you're using only the second
Second one,
ejaculation.
yeah, second one. It will give a good motility. Even we have studied the DNA fragmentation index for the samples. It was very low. It was quite low in the first samples.
Is it counterproductive in people who have borderline or severe abnormalities?
Sorry.
Is the method counterproductive? That means whether the count and motility comes down drastically if we use it in borderline male factor or severe male factor abnormalities?
But first you have to check. But first you have to check the DNA fragment. What is the index? If you can load on it priorly, and after that, this method will help.
So normal spermia with high DFI.
Yeah.
Okay. Thank you, sir.
I heard about a kit which, home-based kit. They can use it home to get their report. It will analyze the semen analysis, everything. Is that
Yes.
practically being used? I mean, any idea anybody has about it?
There's MES has that instrument. I forgot the name, but yes, they are marketing it and they're doing it also.
Yeah.
Any more questions?
Home collection of sample is happening, but home-based test, I don't think so. Bangalore, I haven't heard.
Yeah, you can.
Maybe in
Anyway,
US.
we are not going to use it. We use the backup sample, or secondly, if they are giving and we will usually counsel the patient if it is feasible. Previously, has he given? Is he comfortable? Has he given the second time ejaculation? That patient only will be enrolled in this study.
All
right, thank you, sir.
Any questions in the chat? There's nothing in the no questions I don't see. But Dr. Geeta has mentioned amazing. She's impressed with the talk.
Yeah.
Komal has also mentioned very good presentation by both. Yeah. Congratulations, Dr. Soumya, Dr.
Thank
Seema,
you
and
so
Dr.
much.
Pankaj. We highly appreciate. Nivedita, any remarks? Anything?
I think that thanks for showing us the future.
Yes. Yeah, the videos were remarkable. Soumya, I was really wanting to see those. Also, thank you for enlightening on that website where, if people are oriented towards research, to go to Weiss Harvard, right? www.weissharvard.edu.
Weiss University.
Great. Yeah, I encourage everybody to have a visit once to those sites. I'm sure that is going to be helpful in a lot of ways. I thank Dr. Soumya, Dr. Pankaj, and Dr. Seema for the presentation. We look forward again, possibly in the next year, one more, hopefully. Thank you all the delegates for joining in. It's a fantastic thing to get together every month to have some academic discussion, because that keeps us on the edge, and it's like sharpening your saw to be relevant. At the same time, I have a few announcements. So as you all know, South ISAR conference is coming up. We had requested for some candidates for the quiz. We have shortlisted 3 people. Dr. Arunima, Dr. Chaitra, and Dr. Hemant, who is an embryologist, have formed a team to participate in the quiz. They shall be participating and representing Karnataka or Kisar in the quiz during the South ISAR. I request all the Kisar members to share their map and be present at the time of the quiz. Our next journal club will be in the first week of May. It will be by Gunashila Hospital, Team Gunashila. And yes, it's time to close. I thank you everybody. Thank you, Dr. Nivedita, our president. Thank you, Santosh and other office bearers. Thank you, Soumya, our joint treasurer and the moderator for this session. Thank you, Dr. Seema. Thank you, Dr. Pankaj. Have a nice day. Bye-bye.
Thank you. Bye.
Thank you so much, everyone. Have a nice day.
Put a group photo.
Yeah.
Whoever wants to turn their videos on, they can turn. Official session is
Yes.
over. It's only, nice. So whoever wants to
I request
post,
everybody to.
then we'll take a shot.
Say
One
cheese.
second.
Excellent.
Okay. Santosh, turn on the camera, no? Zakia ma'am is there. Shweta is there. All of you, please turn on and give a very
Dr.
nice,
Shweta.
sweet smile. We are waiting. Waiting. Nice. So shall we?
Navya.
Of minutes 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. How many more counts?
No,
Shall we?
We've had a good attendance, almost 40, 45.
Yeah. Good job.
Which is good for a Saturday evening. Yeah.
Yeah, that's true.
Excellent.
Thank you.
Thank you, guys. Thank you. Bye-bye. Bye.